Hi, this is Heather Peterman. This is week 6 lab, the environmental influences on microbial growth, salt tolerance testing. I've done the usual. I've disinfected my workspace with 10% bleach solution, and then using a permanent marker, I've taken my 6 tubes here, 2 of them with 1%, 2 of them with 7%, and 2 of them with 15% nutrient broth with NaC1 solution, and I labeled one of each with S-Epi, S-CeraVe, and then continued S-Epi on the other ones as well. So they're all labeled here. And then I have my 6 nutrient broths, 5 mLs here. And I have already sterilized the lids, the tips of the vials with my tea light candle here. And I've now placed them in here. So now I am going to take one of my 4 pipettes, and I'm going to put 2 drops of active S-CeraVe culture broth into the 1% NaC1 tube, and I'll do that first. Okay, so now I'm going to put 2 drops of the S-CeraVe culture broth into the 1% NaC1 tube. And then I will repeat these steps for the 7% and the 15%. And I will pass the opening of the newly inoculated tubes over the flame before I put the cap back on. So I will do that for all 6 of my vials now. Okay, so I finished the steps for the S-CeraVe tubes or vials, and then I did the same for the S-Epidermis NaC1 tubes and the active S-Epidermis culture. So I have my 6 completed here. I then took a picture of all of them prior to setting them aside for incubation, and then now I'm gonna place them in my laundry room. That's where that's what I use for incubating my specimens for 48 hours, and then I will check back and then take pictures of my results. So now I'm going to clean up as usual. I'm going to put my pipettes in bleach solution, wipe down my surface with 10% bleach solution, and wash my hands with soap and water.